simultaneously?
Oops. Yes, that is why it is telling you that it's being streamed now. We always double these up. So for those of you on Zoom, we've just started streaming this to YouTube, which is why you should be seeing a content of this meeting is being sent to you, a third-party message. And we are almost ready to get started. It's 7 o'clock. So now,
Bradley, Cecilia, and Joseph are presenting tonight?
yes, and they're all here now. And I'm going to share my screen. So since it is 7, let's go ahead and get started. I'm Vivian New, and I'm going to be your host tonight. And welcome to the first of our research scholarship talks for the year. We have Bradley Sveno, Cecilia Gervin, and Joseph Cuenza, who will be presenting tonight. Sorry, my slide is not doing the right thing. There we go. But before we get started, I wanted to acknowledge that the work done by the Santa Clara Valley chapter of CNPS lies in the homeland of the Ama Mutsun tribal band, the Tamiya Nation, the Ramaytush Ohlone, and the Muwekma Ohlone, who still live and thrive in this area today. We hope to learn from them and support their work to restore traditional practices and to heal from historical trauma. If you're not familiar with CNPS, we are a nonprofit environmental organization we were founded in 1965. And just fresh off the presses, we now have over 13,000 members, not 12,000, but over 13,000 members in 36 chapters that are spread all over California and Baja California. So we are not just in California, we are not just in the United States, we actually go across the border as well. Our chapter is the Santa Clara Valley chapter, and we cover all of Santa Clara County, as well as southern San Mateo County. Our mission is to protect California's native plants and their natural habitats today and into the future. We do that through science, education, stewardship, gardening, and advocacy. If you're not currently a member of CNPS, we would love to have you join us and support our movement to conserve California's native plant diversity. Members get not just one, but 2 journals, Artemisia, which has more scientific articles, and Flora, which has a lot of great general interest articles about native plants. You'll also receive our chapter newsletter, The Blazing Star, which tells you about things that are going on in our chapter area, as well as articles of local interest. And you also get discounts at participating local nurseries and many other things. We have a lot of activities in our chapter. Super simple to join. You can do a sign up online at cnps.org/join. Our chapter has a lot going on. Right now, we have an online plant sale, which will close at midnight. So if you would like to purchase plants, you can do that. You can go to our website, and the link is right there. And that store will be open until midnight. On Saturday, we have a field trip coming up at Villa Montalvo, and that's to look at the California nutmeg tree. It's a very cool tree. If you're not familiar with it, I highly recommend going to this hike. And just go to our meetup site to sign up for it. On Sunday, we have a seed and cutting exchange at the Peninsula Conservation Center in Palo Alto. Great place to get some seeds and plant cuttings to get started. We are moving into planting season, so this is a perfect time to get supplies for free. And if you happen to have seeds and cuttings that you would like to share with others, please do bring them. Next week, on Tuesday, we have our Native Plant ID meeting. That's going to be in Palo Alto at the Peninsula Conservation Center. And this month, we're going to be going over conifers. So if you are interested in conifers or you just want to brush up on them, it's a great meeting to go to. On Friday, we have our photo group meeting that's online on Zoom. And if you're a photographer, you're welcome to join and share pictures. And if you just want to look at pictures and talk about photography, it's a great meeting and is open to the general public. So please think about joining us. We do a lot of restoration and garden maintenance. So we have work days coming up at Lake Cunningham, at Cataldi Park, and also the Native Plant Garden in Kirk Park. And we have that on both this coming Saturday and the following Saturday. To find out more, you can go to our website, cnps-scv.org, or go up to our meetup site, meetup.com, or you can do what I'm going to tell you about on this slide, which is to join our weekly news mailing list. It's a Google group, very simple to join it. You get approximately one message a week, just simply letting you know about activities that are coming up. And you don't have to worry about that long email address. Just go to cnps-scv.org and the instructions are there. As I mentioned before, we have an online plant sale going on right now. It will close at midnight. So if you want to get some plants, you can do that actually even during the talks because this is online. All the proceeds will benefit our chapter. They help us to support programs like this one. And this, I will tell you, since I am the nursery manager, we have a really good selection right now. We have limited supplies of our plants. So it is better if you have an interest in getting some particular plant to shop today rather than waiting for our big plant sale next month. One of the other things you can get at the sale is this new book from CNPS, Wildflowers of California. It's a super field guide, just came out. We have a good price for it. It's lower than you'll get it at the main CNPS store and on Amazon. You can order it and pick it up at one of the places I will tell you about shortly. We also have our big plant sale coming up in October. It will be another online sale, so it will not be an in-nursery sale. You'll have the opportunity to shop online, order your plants between October 11th to 14th, and then on Saturday, October 19th, you can come to Hidden Villa to pick your order up and also to do some other shopping. Coyote Brush Studios will be there. We'll have our usual book sale, t-shirts, there'll be a planting demonstration, talks. It'll be a lot of fun. So again, that's in October. Save the date. These talks that we give are not just me and the speakers. We also have not behind the scenes, but we also have Janet Hoffman, who is here tonight, and she will be taking care of the QA. So if you are watching on YouTube, if you have any questions for the speakers, all you have to do is type them in chat. We'll be monitoring the YouTube chat, and then Janet will read your question to the speakers. So if anybody has questions during the talk, please do not speak up, just type them in chat. We are monitoring chat, and Janet will read the questions to the speakers at the appropriate time. In general, it will be at the end of their talk, but don't worry, just type them in at any point when you have a question. And so, as I assume you know, if you are here, our chapter has a research scholarship program. We've had it since 1999, and the program provides up to $3,000 for both graduate and undergraduate students that are researching topics relative to native plant and native habitat conservation in the San Francisco Bay Area. We are currently accepting applications for the upcoming academic year, and you need to get your applications in by Friday, October 25th, if you would like to apply. Very easy to do that. You just go onto our website, and the URL is right there, cnps-scv.org/research-scholarship, and all the instructions as well as the application are available there. If you forget this URL, all you have to do is remember our website address, which is cnps-scv.org, and there's actually an education menu item that will lead you to this site. Oh, whoops. And so now we are ready for our first presentation by Bradley Speno. He went to San Jose State University. He received a graduate research scholarship for his work with the Alameda song sparrow, habitat use as an indicator of salt pond restoration. And Bradley, I'm going to stop my share and let you take it. So.
Okay, hi. Um, share here. Okay, hi, my name is Brad Spino. Again, the title of my research project at San Jose State is Alameda Song Sparrow Habitat Use as an Indicator of Restoration Success. So a little outline. First, I'm going to talk some about the background of my project, kind of related research. Then my hypothesis and research questions. Methods, which include site descriptions, botanical surveys, and avian surveys. I have some results that are pretty preliminary at this point, and discussion, which is based on preliminary results. So I'm really interested in using birds as a potential bioindicator of restoration success. So in addition to botany surveys to determine how restored a site is, can you use birds as a further metric of restoration success? I think wetland ecosystems are really important, so I focused on South Bay salt ponds for my project. And some of the research I reviewed showed birds as a indicator of restoration success. Mostly the studies I found were in the Central Valley in riparian ecosystems. But as I was searching for Alameda song sparrow information, I found a few studies that they weren't specifically restoration focused. They were more focused on the Alameda song sparrow itself. But one unpublished study from Point Blue described the birds using restored sites slightly more than unrestored sites. And another survey or another study I read said the two were comparable. Neither one narrowed it down to salt ponds specifically. So I thought I would answer or try to answer that question. So the Salt Pond Restoration Project, for those not familiar, is a restoration project in San Francisco Bay where former salt ponds have been either for the purposes of my project, I focused on the salt ponds that have been open to tidal water. So the goal is for them to become tidal marsh again, which is the preferred habitat of the Alameda song sparrow. A few studies show that the subspecies of song sparrow are specific to the salt marsh as opposed to the upland song sparrow, which is more specific to freshwater sites. The subspecies is a California species of special concern per the California Department of Fish and Wildlife. And as I said, it's one of about 24, 25 subspecies of song sparrow on the West Coast. So my questions hypotheses, my null hypothesis is that there's no relationship between the percent cover of native plant species and Alameda song sparrow abundance. Start with my null, see if I could disprove that. Then my research question one is what the relationship is between the age of a restoration site and Alameda song sparrow abundance. I'm defining age as the time since the breach of a particular levee. So how long has the site been tidal? That's what I'm defining as age. Then I have a question specific to habitat conditions, including levee width, proximity to channels, percent cover of dominant plant species, and vegetation shape, so size and density, are most indicative of nesting song sparrow abundance. And then I finally have a question about how the age of restoration might affect the percent cover of native plant species. It's a little bit more complicated because certain sites of mine have been restored manually and some have just been untouched. because the song sparrows nest in the upland transition zone, kind of an ecotone between the marsh and the levee, the upland may or may not be as restored as the marsh itself. So here are my sites. I have 7. They're all across different age of breach, and some are historic. So I have E9 up in Eden Landing, Hayward, which is breached in 2011. Island Pond A19, which is breached in 2006. It's just out of Warm Springs. Alviso Pond A6, breached in 2010. Outboard Charleston Slough, which is a historic site. It's also shares a levee with A1. Faber Laumeister being historic, Faber being breached in 1971, my oldest site by far. And Inner Bear Island up in Redwood City, which is breached in 2015. Here's some more information on my sites. Again, some of them have been restored, some of them are less restored. There's been different success of each restoration. They're all fully tidal, some muted tidal, which just means the tide water comes in a little slower. They're all different sizes in hectares, but a little bit more comparable in the approximate levee length, accessible at least, the part that I could walk. So into my methods, I divided each of these walkable levees. I used 2 to 3 randomized 200-meter transects with 10 quadrats within each transect to measure percent cover of plant species, vegetation height, transition zone width, slope, aspect, and notes on water features. I also, I'll get to it in a minute, I established mist nets at 4 of my sites. So I put 2 quadrats per 12-meter mist net length, and I randomized those as well. Then I combined my mist nets into a banding lane. And then I surveyed twice, once at the end of 2023 growing season in September, October, and one mid-growing season, 2024, May, June. And my avian survey methods, I then walked each transect and each banding lane for 20 minutes each, and I counted every song sparrow I could find within that time. And I also tried to estimate whether it was a adult or juvenile. I also took notes on whether I thought it was a male based on whether it was singing or not. I determined whether they were juvenile or adults based on how many in a group were foraging together. Typically, I was only able to see this during the breeding season when I expected family groups to be foraging together. I surveyed 3 times, once in the fall of 2023 after the breeding season and after the expected molt period for the species, and then twice in spring of 2024, spring to summer of 2024. And then, as I described, I mist netted and bird banded at 4 of my sites. If anyone is unfamiliar with mist netting, this right here is a picture of a mist net. This pole holds it up. It's basically a volleyball net that catches birds. And then we pull them out and put at least a metal band on them. We tried to put we put color bands on most of what we captured. Some escaped before we were able to do that, but the color band application helped me to go back and recite the birds after we captured them originally. So during my song sparrow counting period, I would go back and try and estimate or try and find the color bands to identify individual birds. So as I mentioned before, I have pretty preliminary results. So far, I've just done a couple of novas and a few box plots, but this was a pretty significant difference here. I had a p-value in my ANOVA of native species richness per site. So here are my 7 sites down below, and my native species richness up here. So I have labeled groups A and B, where there's a significant difference between these groups and this group in terms of native species richness. And to describe my botanical survey results a little bit further, I had 75 total plant species and 17 defined as dominant, which I defined as over 50% in total coverage within a quadrat in more than 5 quadrats, because I didn't want to count a plant that was only dominant once or twice in a quadrat. I figured at least half a transect would be appropriate. So 7 of my dominants were native, and 10 of them were non-native, unfortunately, a bit more of a skew towards the non-native species. And I'm currently working on a PCA, which is a big graph where all of my variables go in. I could find more trends such as this. I'll also be putting in my bird count results. Right now, I've just put these in by site, not by transect, but I was unable to find a significant difference between any site in terms of Alameda song sparrow abundance. This is just birds per surveyed count. I'm also curious to see if there's a difference between the non-nesting season and the nesting season. So I will be running that analysis as well. My banding results are kind of interesting. I did see a trend towards the older, more historic sites in terms of captures per net hour. So we had about 5 nets open at any given day. Some 3 of my sites, everything except for A19, we did for 2 days. A19 I did just 1 day. So I standardized my net hours and to standardize these numbers. So again, I haven't run an analysis on this yet, but I am hopeful to see a significant difference. It's also interesting because in Island Pond A19, where the vegetation was at this point anecdotally densest, I had the highest diversity of birds. I have similar species in other sites, such as Faber and Eden Landing, but at A19, I had much higher numbers of all of them and fewer numbers of Alameda song sparrow. So I thought that was pretty interesting. Here's just some photos of each bird. This is the Alameda song sparrow here. You can kind of tell it's a little bit yellower in the underparts. It's supposed to be slightly yellower than an upland song sparrow. I didn't find that in most birds. Maybe 1/3 to a half of them were noticeably yellower, as compared to a savanna sparrow on the left here. It's got the yellow eyebrow. Then I had a nice marsh wren over here, common yellowthroat, and we even caught a cowbird, a brown-headed cowbird, which is kind of fun. Oh, and right here we have a cliff swallow. So as I said, there's little difference in the bird counts across each site. One group of 4 in terms of plants was significantly higher in richness than the others. So there's a potential lack of preference for native plant diversity, meaning the song sparrows probably can't act as or might not be able to act as a bioindicator for restoration success. But like I said, I have more analysis to run. And as I described, the highest concentration of other species at A19 was we observed, and the vegetation was most dense there. If I just put this picture in to kind of show A19 as very dense, very much mustard, and nothing really else in the upland, at least. In other sites, this was a full road with two tire tracks. This was A19 was more of a bushwhack. But yeah, I just want to move on to my acknowledgements now. Thank you to my committee, Dr. Lynn Trulio, Dr. Rachel O'Malley from San Jose State, Dr. Katie LaBarbera from San Francisco Bay Bird Observatory. She helped me. She was actually my supervisor for my banding, so that part of my project would not be possible without her. Um, Grant organizations, California Native Plant Society, thank you, and the San Jose Kiwanis Club. Other SABBO staff for providing support in my project. San Jose State cohort and volunteers made it a lot easier. Family, friends, coworkers, thank you. And if you have any questions, let me know. I'm not sure if we're doing them now or after all of the talks, but here are then my references.
We could take questions now, but I don't see anything in chat yet. I don't know if Vivian has anything either.
Okay.
And nothing on YouTube either.
Okay. Well,
Okay, thank you.
Thanks, Brad. Actually, I did have a question. I was wondering, do song sparrow, the Alameda song sparrow, is it primarily a seed eater, or does it also eat insects? Is there a
As I understand, they eat a lot of seeds. I saw pretty they do need to eat bugs for protein, insects, bugs. Um Primarily, I'm not sure. I did read a study about pepperweed, lepidium, how its presence, I think, kept the song sparrows at a site longer than it would have normally. As I remember, I need to read back.
I was just wondering because, there's all, I don't know if you know who Doug Tallamy is, but he's a professor of entomology at University of Delaware, and he's done a lot of work around these relationships between native plants and, well, in urban areas, actually, if it makes a difference. And uh, one of the things he often brings up is the need for caterpillars for success, reproductive success, because baby birds can't eat uh, the, the seeds. And so I was just wondering if you had seen anything around, the nuts versus whether or not nuts or native plants, and because native plants often correlate with more caterpillars. So that was just curious. Thanks.
Right. No, that's interesting. I No, I mostly just saw, again, anecdotally, but it might come up in my analysis that the denser plants, no matter whether they were native or non-native, just provide more cover for the birds, for the song sparrow specifically.
Thanks.
Thank you.
All right. So now I'm gonna go to our next talk is going to be by Cecilia Gervin from UC Santa Cruz and Joseph Cuenza at UC Santa Cruz as well. Cecilia is a recipient of our graduate research scholarship, and the title of her work was The Role of Sexual Conflict in Hermaphroditic Floral Evolution. Joseph worked with her, and he received one of her undergraduate research scholarships, and he was looking at the role of plasticity and pollinators on the expression. And so, Cecilia, I will turn it over to you. I'm going to stop my share, and if you can.
All right, let me share. There we go. Oop, let's see.
I just I'm going to spotlight those images. And so you should be able to share.
All right. Is that, wait, no, I have not succeeded. Okay. How is,
Your whole desktop is showing up right now. Is it?
okay, that's not what I want to do. Okay.
Perfect. That looks good.
Alrighty. And have I gone full?
I think it is full screen. It looks full screen.
Okay. Okay. Alrighty. Um, so I'll be talking about conflicting selection and floral evolution in Clarkia unguiculata. And I'm Cecilia. I'm starting my third year as a PhD student in Dr. Kathleen Kay's lab at UCSC. And I want to talk to you about the work I did this summer with CNPS funding and how that fits into the rest of my dissertation work. And Joy, I don't know if you want to hop in and just say hi.
Yeah, hi. My name is Joseph Casenza. I also go by Joey, and I was an undergraduate student and researcher in the K Lab, and I will be talking a little bit about my contributions to the project as well.
All right. Sweet. So a little bit about me. I graduated from Cal Poly in San Luis Obispo in 2020 with a bachelor's in biological sciences. There I was lucky enough to be mentored by Doctors Jen Yost, Matt Ritter, and Dina Grossenbacher in various plant ecology and evolution projects, from whom I got a really well-rounded plant science education. Through research experiences I had as an undergrad, I developed an interest in the complex world of plant reproduction. And I did some unrelated work after, or like I did plant related work, but not plant reproduction work. After I graduated, I went to OSU in Oregon. I had a fun summer working as a tech. I did plant ID. This was a lot of like differentiating between Ammophila species. So after I had enough of ligules, I worked at OSU Oklahoma with Dr. Eric Lopresti and helped with some experiments and projects looking at seed functional traits. And then eventually I made it back to plant reproduction in Dr. Kathleen Kay's lab, where I was first a field tech helping with her Costus project. And there's a photo of a little Costus floral visitor in the bottom left. And now I'm a PhD student with her in my third year, and I'm working in one of her other systems, which is Clarkia. All right, so I started grad school knowing that I was interested in floral evolution and how pollinators can mediate that. And in the past 2 years, I've become fascinated by the potential role of conflicting selection in floral evolution. Specifically, I want to investigate how pollinators can mediate conflicting selection on floral traits, given that male and female fitness is achieved differently. So conflicting selection is what it sounds like. It's basically what happens when one selective force is at odds with another. It's a common evolutionary phenomenon, but it's hard to study unless you're able to parse out the effects of a single trait on different components of fitness. In a typical bisexual outcrossing plant, which is the majority of angiosperms, as I'm sure you are all aware, plant fitness is composed of two parts: female fitness, which is determined by the number of seeds produced, and male fitness, which is determined by pollen export and the consequent number of seeds sired. I'm working with Clarkia unguiculata, or the farewell to spring, so called because it tends to flower at the onset of California's hot, dry summer. This species is a widespread and common roadside annual endemic to California, whose range spans a dramatic east-west climatic gradient from the hot, dry Central Valley to the cool and damp coast. The genus has been widely used as an evolutionary study system due to variation in mating systems. Clarkia unguiculata has a mixed mating system where it mostly outcrosses, but is capable of selfing, and geitonogous selfing or selfing between different flowers on the same plant may be fairly common in plants with larger floral displays. The species is pollinated by many bee species, but they do have a special a specialist pollinator, Hesperipus, which we'll talk about later. And visiting bees are either collecting nectar or collecting pollen from the flowers. All right. So Clarkia can have many flowers in their lifetime, and each individual flower can be open anywhere from 1 to 11 days, during which the anthers release pollen in 2 whorls. This gradual release of pollen increases male fitness via a mechanism called pollen dosing, which is termed by my advisor. And there's a considerable amount of variation in how long the male phase can last, both in a common garden and in the field, as I have measured. And so here you can see the first whorl of anthers, the inner anthers, and the second whorl of anthers. And there's some interesting, like, as you can see in these photos, the pollen is different colors, which isn't always the case, but it's just a very fascinating trait. And then after the male phase, or typically after the male phase is completed, the stigma lobes will open and the female phase will begin. There can be some overlap, and it's possible for pollen from the very same flower to pollinate the stigma. So, and the amount of time an individual flower is open is what I'll be calling floral longevity as I continue to sort of set up the theoretical part behind what we did. So because seed production is water costly, female fitness is typically limited by water. Clarkia can lose a substantial amount of water through soft floral tissues, and long floral longevity has been shown to come at the expense of seed production. Thus, the female optimum would be to keep flowers open just long enough to receive enough pollen to fertilize all ovules and therefore conserve water for seed production. Which is to say, if they're open longer than every ovule is fertilized, it's just a waste of time from like the female perspective in theory. Male fitness, on the other hand, is limited by access to mates, and quantitative work done by my advisor has shown that by releasing small amounts of pollen onto pollinators over a long period of time, Clarkia can achieve greater male fitness. Longer floral longevity is thus likely selected for by male fitness via the advantages of pollen dosing. Though it's important to note that this is only an effective strategy when there's high pollinator visitation, which is to say, like, if a flower can only expect to get 1 visit in its whole lifetime, it doesn't make sense to dose a small amount of pollen. You might as well put all your pollen onto that 1 visitor. All right, so if the female optimum would be a shorter lifespan and the male optimum would be a longer lifespan, there's clearly going to be conflict. However, this conflict is context dependent. For instance, in the absence of water stress, longer floral longevity might not come at the expense of seed production. And additionally, if pollinators are scarce, not only will pollen dosing not work, female fitness might benefit from keeping flowers open longer to increase pollen receipt, thus reducing conflict. All right, so Clarkia's eastern range is hotter than its western range. This results in greater water limitation and also greater predicted pollinator availability inland, since bees tend to be more abundant and active in warm, dry environments. In my project, I will leverage the concurrent climatic and pollinator availability gradients that it spans to have access to populations that are both in high predicted conflict and low predicted conflict. But is this really like the case? Do we see greater limitation on the coast? If yes, why? That's kind of the main question of the summer. This summer, I used a pollinator limitation experiment across Clarkia and Guiculata's range to test if some of my populations are under greater pollen limitation than others. This is critical for other components of my dissertation, since only in the absence of pollen limitation can a shorter floral longevity benefit female fitness. Secondly, I wanted to know if differences in pollinator assemblages and/or abundances could explain variation in that variation in pollen limitation. And thirdly, I collected a whole bunch of seeds from my populations for future experiments that'll be starting soon in the greenhouse. All right. We worked at 3 sites this year. Pinnacles National Park was our hottest, driest, and most eastern site. Hastings was our intermediate site, and Big Creek UC Reserve was our cool coastal site. In the future, I hope to work at an additional pair of inland and coastal locations further north, hopefully closer to Santa Cruz. But it was difficult to make that work this year. All right. So to achieve my first aim, I conducted a pollen limitation experiment at all 3 sites. I selected a large population and marked 30 random plants that had at least 3 to 4 fresh open flowers. And for each plant, I marked and manipulated up to 4 flowers. 1 flower was unaltered, 1 I emasculated by removing all anthers. For 1, I added supplementary pollen. And treatments A and B were done on flowers that had just opened, and treatment C was done on flowers that had just begun their female phase. For a subset of plants, I emasculated and bagged the flower as a methods control. And obviously, ideally, they would have been flowers like at exactly the same time, but unfortunately, because they do have that, they are dichogamous and they're male first and female second, I couldn't do all the treatments at the same time on flowers that were more or less the same age. They had to be spread out a little bit. So here's some pictures of what those treatments looked like. I marked flowers with a small label directly below, and you can see that tag in this unaltered treatment A, which provides the seed set under natural conditions. This is treatment B. I would sterilize the scissors in between plants to reduce the chances of transmitting diseases. And the resulting seeds from this treatment would be seeds resulting from only outcrossed pollen. It doesn't, gytonogamous selfing could also be happening, but for most of the plants, they don't have so many flowers, and the chances of a bee bringing pollen from another plant is pretty high. And here is a sort of shabby looking treatment C. I would typically try to pick a fresher flower, but that sometimes wasn't possible for small plants that had few flowers, and especially locations that was very difficult to reach plants. And so this wasn't always the case, but unfortunately, it's the only photo I could find. But I would use either a pipe cleaner or a toothpick to transfer pollen. And donor pollen was a mix of pollen from 2 to 5 different plants from the surrounding area. So I was trying not to pick, something that was directly next to it that would potentially be a sibling. All right, and then here is a treatment D, bagged and emasculated treatment, which is just a check that my emasculation treatment was working and that the plants were not able to produce seeds apomictically. To collect seeds, fruit tips were taped 2 to 3 weeks later, and seeds were collected after fruits began to split. So it's necessary to tape the tips because Clarkia fruits will dehisce from the top. And we use clear tape to do this because fruits will continue to photosynthesize as they develop. All right, and currently we are in the seed counting process. So unfortunately, I don't really have any fantastic results to share with you as of today. Though based on what we have done so far, it does look like all of our treatments were working, like things that the treatment C with supplementary pollen tended to have greater seed set than the treatments B and A, and that kind of thing. So I feel good about that, but I can't say anything too much about the comparisons between sites. All right, and now for our second objective of describing pollinator assemblages, I'll hand it off to Joey, who is an undergrad researcher in our lab, as he mentioned, and received funds. And he's been a great help with the pollinator observations and the bee trapping, and has been really spearheading the bee identification, which I'm for sure not an expert in at all. So, all right, go for it, Joey. And I will just advance for you.
Cool. All right, so a little about me. I recently graduated with my BS in environmental science from UCSC. Over the past year, I've been working in the K Lab, helping Cecilia with her project. During my time at UCSC and in working in the lab, I took an entomology course that renewed my interest in insects. And I'm interested in potentially working with insects in the future and exploring opportunities in that field. All right, so my role in the project was to conduct pollinator observations to document the variety and behavior of insects visiting flowers. I recorded data such as the time of day the insect visited, the type of pollinator, to the best of my ability based on what I could see through the binoculars I was using, and how it interacted with the flower, whether it was collecting pollen or if it was nectaring. I also set out bee bowls filled with soapy water to hopefully attract and capture the insects that were visiting the flowers for future identification and to potentially get a measurement of abundance. I would occasionally use a net to capture some insects that visited if I noticed that they were not falling into the traps. The identification work is still ongoing, and I'm using various identification keys and other resources like iNaturalist to determine genus and, when possible, species, but pretty difficult to get to species. Here's a chart that shows the most common visitors that I saw at each site in my observation periods. We defined visitors as insects that could be seen making contact with the plant's reproductive parts. In the next slides, I will discuss the most abundant visitors per site. Species identities are being worked on using specimens collected from the bee bowls or from netting. I would also like to note that there were honeybees in Hastings. They were observed in Hastings, but they just weren't as common, and I didn't see them visit Clarkia, so that is why they're kind of missing from that Hastings tab there. Now, the most common pollinators in pinnacles were the honeybees, Apis mellifera, and the Clarkia bees, Hesperapis regularis. And the most common pollinators in Hastings were small mason bees in the genus Hoplitus and small carpenter bees in the genus Ceratina and Clarkia bees again, Hesperapis. And then finally, the most common pollinators in Big Creek were honeybees, mason bees in the genus Osmia, and again, Clarkia bees. So Hesperapis regularis, the Clarkia bee, is a native bee and a Clarkia specialist, and was anticipated to be prevalent at all three research sites and was observed at all three sites. But interestingly, it was not always the most common pollinator. At Pinnacles, the western honeybee exceeded the Clarkia bee in visitation frequency and was a close second in visitation in Big Creek as well. These findings highlight the dominant role of non-native honeybees in pollinator communities, even in habitats where native specialists are expected to thrive. Another interesting fact was that despite the honeybees' dominance in visitation, they were never caught in the bee bowls at any of the sites. Now the Hoplitus bees were interesting because while they were common visitors, they were captured in the bee bowls many more times than they were seen visiting. So this can suggest that bee bowls may disproportionately attract certain species, making them less reliable for estimating true pollinator abundance. These findings highlight the limitations of relying solely on passive trapping methods like bee bowls and the need for maybe other methods like netting to get accurate pollinator estimates. Now, for my conclusions, honeybees were prevalent across all the sites, not as commonly observed in Hastings, but notable there, and often outcompeted native species like the Clarkia bee in visitation frequency. And although Hesperapis was expected to dominate due to its specialization on Clarkia, it was not always the most common pollinator, raising questions about competitive dynamics with honeybees. Then there was the Hoplitus species that were most frequently caught in bee bowls, but were likely overrepresented, which could show a limitation of passive trapping methods for estimating true pollinator abundance. And I would also like to give a special thanks to the Santa Clara CNPS chapter for their support and funding, which made my involvement in this project possible.
Sorry. I would also like to thank the Santa Clara Native Plants or CNPS chapter for their support, and it's this summer was a great foundation for what I'm going to build the rest of my dissertation on. I've got the seeds for greenhouse experiments and have my sites for a field experiment that I'll do as well. And I'm hoping to do 2 to 3 more summers of pretty much what we've described to get a better idea of pollinator assemblages and abundances in both through space and time. And yeah, that's it. And also would love questions or comments and all that. Alrighty, thank you.
Okay, there are couple of comments. Brad Spino says, very cool. I didn't know there were bees specific to Clarkia. And I can echo that comment myself. I didn't know that either.
Yeah, they're awesome. I should have put a photo in, but the male bees will be like Clarkia cylindrica, which is often concurrent with unguiculata, will roll up at night and you'll see the little male bees inside the flowers. It's really sweet.
I'm gonna go have to take a closer look at my Clarkia. Thank you. And Carolyn Dorsch says she's interested in the outer and inner whorls of the sepals. Do the same flowers have both? And if so, are they present at the same time?
Inner and outer, let's see.
I think
Oh,
you
gosh.
talked about 2 sets of stamens.
Yes, 2 sets of stamens. Yeah, so all flowers have 8 stamens total, and there's a smaller inner whorl that as soon as the flower opens, those begin to dehisce. You can see that on the left. And then once those have kind of shriveled up and are done, the outer whorl, which was previously reflexed, will move to the center and begin to dehisce. So right in the center of the flower is where the bees will interact with the parts.
So the inner anthers release the pollen first and then the outer anthers?
Mm-hmm.
But they're both present at the same time.
Yes, they're present at the
they
same time.
mature differently.
You can see in the first photo on the left, those pink like canoes are the outer, is that second whorl of anthers that just haven't opened yet.
Okay, and she wanted to know if the Clarkias in the eastern populations have differences from the same species in the more western and cooler habitats.
They definitely do. The sort of like maybe what you would expect for a more desert, locally adapted plant. The leaves are thinner, the plants are smaller, flowers are a little smaller.
And also want to know, do bumblebees visit any of the Clarkias?
Yeah, I can. We did see some bumblebees visiting Clarkia very infrequently or uncommon, but they were there. I've only gotten one. We only caught one. I didn't ID it yet, but they were there, just very rare occasions.
Okay. And then Jess Goldenberg wants to know a little bit about your plans for the seeds in the greenhouse.
Okay, so, oh gosh, I have like, rather than do transitions, I just have like 1 million slides. So here you can see the greenhouse setup, and we did a pilot of this this spring, but we'll submerge the cones in different depths, and we're going to mimic a drought. We'll do a drought experiment in the greenhouse, and we're looking to see if the eastern populations are able to be plastic with their floral longevity. So if the benefit of floral longevity is greater inland, but also the potential cost is greater, then when they are exposed to drought, are they able to shorten their floral longevity is what we're looking for. Hopefully that made sense.
Yes, okay. And Jess also wanted to know, was there any pollination by other insects such as flies, butterflies, or moths?
Yeah, actually, no butterflies or moths specific to Clarkia, but there were flies. Very, very so often there was a fly, but well, I wouldn't say they were pollinating really, but they were there and they would visit and they would take nectar from the flower, but they wouldn't touch any reproductive parts. Not good pollinators, basically.
Hmm.
Not like the bees.
Hmm.
Yeah.
There are some Clarkia species. Clarkia breweri is a hawk moth specialist. So some Clarkia species will have moths, but not unguiculata.
Okay, and Marianne Damon says, very important implications of your findings on the insects. Great field work. Compliments to you. And then
Thank you.
Vivian, did you have any on YouTube? Any questions?
Nope, YouTube is quiet.
Okay, well, thank you both. It was a very interesting presentation.
Yeah, thank you.
Yeah.
Thank you.
Okay, so I think that wraps us up for the evening. I wanted to thank Brad and Cecilia and Joey. Really great presentations. Wish you luck in the rest of your research. We'd love to hear back when you're finally done. If you would send us a copy of your dissertation, we would love to have that for all of you. And thank you all, the audience, for showing up. And we will have more talks in the future. We have more scholarship talks coming up. I think our next one is on October 3rd. So I hope to see some of you there then. And I think that wraps us for the evening. So thanks again, and have a great rest of the evening, everybody. I'm going to end the Zoom session now. Good night.
Thank you.
Yeah, and thanks, Janet, for